Question number 133. Number of plant species reported in India. As far as the biodiversity, if you are clear in the biodiversity part, right, if you are good in this particular chapter, you can very well answer this type of question. The number of plant species are reported as if the record says it's 45,000. 45,000 is the correct answer. So, option 4 is the correct answer as per the facts says, but it's not. But now if you go and do the research again, definitely it cannot be 45. This would have been rapidly or increased, or it might have been decreased. These are subjected to changes. So, option 4 is the correct answer. Question number 134. Zygospores are formed in, where you could see the zygospores which you could see in case of mucor, and you could see in case of rhizopus. Because in mucor and rhizopus, you could the structures, the zygospores, it gets usually produced at the time of sexual reproduction, right? So, penicillium and all, you could see the conidiospores usually get produced. So, mucor and both will resemble same. For question number 130, you could see 134, the mucor and penicillium. So, what makes the difference? Like you could see the mucor structures which will be like this, and sometimes the mucor will be having some branches like structure. Sometimes you could see the branches also in case of mucor, but no rhizoids. But you could see in case of rhizopus usually will be having the structures, and you could see this type of structure, but the rhizopus will be having the rhizoids. The rhizoids you could see, and no branches that you could see. Here you could see the columella part that you could see. But mostly like rhizopus and mucor, both will be exactly same. This is a sporangium part, and the inside the sporangium you could see the sporangiospores. This is mycelium. This is sporangium, and this is a rhizoid, right? And you could see this is a columella that you could see. There are very slight difference only that you could see in case of between the mucor and rhizobium, that usually perform the stain called lactophenol cotton blue. Lactophenol cotton blue is the stain that usually perform for this type of mold to see under the visualize under the microscope. Fine. So, question 134, option 4 is the correct answer. 135. In atoms, protons and neutrons are present. Where these are present? It's present only in the nucleus. So, option 135. 1 is the correct answer. 136. Keystone species are species that determine the structure of the biotic community. So, option 1 is the correct answer for question number 136. Okay. Question 137. The science of naming the plant is known as how we call it as nomenclature. It's a nomenclature. Where you could see the nomenclature, where it's a name, it's a nomen means what? It's a name. Clature means to call, okay, to give the distinct scientific names to various structures, including living organisms, so that they are easily identified. So, by this help us to to avoid wasting our time by doing the same sort of research with the existing organisms. So, it's a naming of the plants, that is nomenclature. Question number 138. In nostoc, the heterocysts are intercalary. In case of nostoc, that you could see, it is intercalary. In between the other type cells, you could see the presence of this particular heterocyst, and this heterocyst which plays an important role in fixing that atmospheric nitrogen, where this carries like nitrogenase enzyme which requires anaerobic environment, because the nitrogenase enzyme function better only in the anaerobic condition. So, for this question, that is intercalary is the correct answer. 139. The branch of botany concerned with the classification, nomenclature and identification of plants, that we call it as systematic botany. This for the question 139. Option 2 is the correct answer. Question 140. Causes of mimicry. Mimicry is, it's by protection as well as for the isolation. The animals start mimicking, right? For what reason? Usually the animals which will not give a mimic, it's not during in case of attacks, it will not mimic. Only in the case of protection and case of like an attack only, what happens? Like this will mimic. But when it going isolation, like isolations, it will not mimic. Usually in case of protection and the attack only, this type of mechanisms, that is mimic mechanism, that which can be appearing. Because protective mimicry, where the animals imitate other animals, the natural objects, in order to protect themselves from what? From the predators. Predatory animals mimic sometimes natural objects so that they prey get confused and it easily get caught, or attacked. This type of mimicry called as aggressive mimicry. So mimicry is by during the case of protection and attack. So option 1 and 3 is the correct answer for question number 140. Question number 141. Glycogen is most structurally similar to starch. In plants we call it as starch, in animals that we call it as a glycogen, the reserve materials. Question 141, starch. Because starch common in glycogen in animals, that we discussed as the difference between the plants and the animal cells. Question 142. The reduction division in the life cycle of rhizopus occur at the, we discussed already, at the germination of zygospores. This produce the rhizopus which produce zygospores, which produce zygospores. Okay. So question 142 at the time of germination of zygospore. Question number 143. Autogradiography in statement 1 involves that radioactive isotopes like C14, P32, S35, etc. Statement 2, that is in H3 in thymidine, was employed by Taylor for studying chromosome replication. Both statement 1 and 2 are true, both are true, but statement 2 is not the correct explanation of statement 1. So for question number 143, because autogradiography is which is used to study the synthesis of molecule and to trace actually the metabolic pathway of the events. For that only use autoradiography, right? And it involves incorporation of radioactive molecules which usually get labeled onto the surface of the probe. What is a probe? Probe is nothing but a short oligonucleotide sequence. It's same like that of a primer, but if you attach this radioactive label substance, then that we call it as a probe, right? And now what happens at the detection of photographic technique, like we just pass the illumination onto this surface, and then what happens, it start illuminating, the isotopes would start illuminating. The most radioactive isotopes that employed in autoradiography are beta emitters like H3, C14, and P12, Ca14, I131, Fe59, and you could see S31. And this also, autoradiography was also utilized by J.H. Taylor and co-workers for the study of the duplicating chromosomes in the root tip of a Visia faba. So this type of plants where they used. So both statements are correct, but statement 2 is not seems to be correct. The correct justification for statement 1. Okay. So question number 144. The disease produced by fungus Ustilago, are known as smuts. Because what is the reason for that? Because they produce sooty mass of the spores. So sooty mass of the spores, because of this reason, the produced by fungus Ustilago are called as smuts. So option 1 is the correct answer. Question number 145. 145. When you see the statements, a potted plant, that is statement 1, placed in a window shows a bending of a stem towards the light. Okay. And statement 2, this is an example for teleology. Both statement 1 and statement 2 are true, but statement 2 is not the correct explanation of the statement 1. Because the reason is light stimulates, definitely the light stimulates, because the plants will respond to the sunlight. So the plants will bend towards the light. This is correct for different parts in a specific direction. The plants definitely it migrates or move. But it is called as phototrophism. But on keeping the potted plant near an open window for dark room, we observe the plant bending towards the light source as a result of phototropism. Then teleology involves explanation of the natural phenomenon. It's explanation of natural phenomenon in terms of a need of organisms, like the stem bends towards the light because they need a light for the growth. For this, we go with the option 1. Question number 146. Barophilic prokaryotes. Baro means pressure, right? The barophilic, it's a pressure loving organism. Whenever the philic which comes, like it turns love, the love towards allophilic, barophilic, thermophilic, right? Barophilic means it love towards like towards a pressure. These prokaryotes, the bacteria, usually these comes under the Archaebacteria groups, where it grows and multiplies in very deep marine sediments it can grow. So option 146, you go with option 3. 147, identify the correct sequence of the taxa in the linear hierarchy. Class, order, genus, species. Okay, first we have the class, order, genus and species. So question number 147, we have class, order, genus, and we have a species, and then the species we have the varieties. We have a subspecies and we have the variety. Okay, before this only we have the kingdom, the phylum, everything which comes. This is the common hierarchy we talk about. So for this question, option 1 is the correct answer. Question number 148, which one is not a category? Category, class, species, phylum, kingdom, these are all like the category that we giving it. But this one you could see, Glumaceae, it's not a category. So question 148, option 1 is the correct answer. Question 149, which surgical instrument are boiled in water before use? Why? The reason why, it's this process we call it as sterilization. This process we call it as sterilization. We have different types of sterilization that we use, that autoclaving, autoclave technique. We have autoclave. We use 121 degrees centigrade for 15 minutes for autoclaving. Same like the pressure cooker, hot air oven, right? We use 140 degrees centigrade for even 45 minutes that you can use it here. And we have incineration. Incineration is completely destroying, burning, right? Incineration, burning. Then we have fumigation. These are different types of sterilization that we perform. Fumigation for the closed environment like operation theaters, theaters like a closed environment. Incineration is burning, right? So why we are actually doing this? Surgical instruments are boiled in water before we use, for killing the pathogens. If there is any destroying an organisms, like if nowadays like they're not boiling it, now they're doing it what? They're doing with the autoclave. Autoclaving the surgical, whatever things used for operation theaters and for surgical things, either with autoclave or with the hot air oven they can use it. So question number 149, 2 is the correct answer. Question 150, a substance that causes the disintegration of the bacteria. It's bacteriolysine. Bacteriolysine is the correct answer. It lies or kills the bacteria. So 4 is the correct answer. 151, which of the following life cycle is associated with the mucor? Mucor, you could see haplontic cycle. So just majority is what? Is the haploid. Only limited part, minor part is only the diploid. Majority of the life cycle that you could see in mucor, it's of the haplontic life cycle. So for this question, 2 is the correct answer. 152, in 5 kingdom classification, which kingdom incorporates prokaryotes? It's very simple, Monera. Monera is for the prokaryotes. 3 is the correct answer. Question number 153, antibiotics are mostly obtained from actinomycetes. Question number 153, actinomycetes. It's a bacteria where you approximately could see 2100 antibiotics so far they identified or discovered only from this particular bacteria, that is from actinomycetes itself. Okay, so question 153, 2 is the correct answer. Question 154, why flora, manulas and monographs are included in the taxonomical aids? Because for both the reasons 1 and 2, because they provide the index of the species found in the particular area, and we have because they help in the correct identification. So option 4 is the correct answer. 155, fungi differs from algae in all the things where you could see the 4 options, without chlorophyll and possessing chitinized wall, because fungal cell wall is made up of chitin, right? And there is no chlorophyll in the fungi, right? So with this we go with the first option. 156, that is the reason why the 5 kingdom concept, there is no separate kingdom for algae, because algae falls under the kingdom Monera, algae comes under Protista, algae even comes under Plantae. All the 3 kingdoms where you could see like some plant like features also you could see in algae, but fungi is entirely different. Okay, so question 156, in the 5 kingdom concept system of classification, which single kingdom following can include blue green algae, nitrogen fixing and methanogenic bacteria? Monera. So option 2 is the correct answer. Then 157, Unlike other algae, diatoms do not readily decay because diatoms, the cell wall is made up of silica wall, silicaceous wall, that we call it as 157, which is made up of silica wall in case of diatoms, and this silica wall usually which will not be degraded. So what happens, this will be get deposited at the bottom, right? Then what will happen again, it will be piled up, and when piled up it will be keep on increasing, and this forms a structure, the silica wall, which forms a structure called diatomaceous earth. This we call it as diatomaceous earth like structure. This is the feature of the diatoms. This we call it as, I'll write it here, diatoms. Fine. So we're done with this question. Next, 158. Which of the following is a trace element that you could see in a small amounts in most living organisms? Very small. We study macro elements and micro elements. You could see iron, but hydrogen, oxygen, nitrogen, this is the macro elements, but iron is the micro element. There you could see the iron is responsible for hemoglobin formation, RBC, and its transportation of oxygen is required, which is like 97 to 99%, and you could see the carbon dioxide is of 23%. So iron is a trace element. Question number 159. The correct sequence of stages of the growth curve of the bacteria. As far as the growth curve of bacteria is concerned, question number 159. When you plot the growth curve, so how it will come? It comes with the lag phase, log phase, stationary phase, and during unfavorable conditions go to the death phase. This is a lag, this is log phase, this is exponential phase, this is a stationary phase, and this one is your death phase. Okay, in the normal case, in case of the bacteria, this is how the sequence come: lag, log, stationary, death phase. The death phase can also be called it as a decline phase or decline phase. Option 1 is the correct answer. 160. Circular DNA molecules occur. Circular DNA, we are talking here about the plasmid in the bacteria, that is like chloroplast, and also in the mitochondria. If it is in bacteria, we call it as a plasmid, but it is present in chloroplast as well as mitochondria. Mitochondria also is carrying its own extra, that is like its own mitochondrial DNA. Option 1 is the correct answer. Then question, we go with the next part, is 161. The smallest particle of water is a molecule. So option 4 is the correct answer. Question 162. The recent branch of taxonomy is karyotaxonomy. There is a nucleus, karyotaxonomy. 163. Paramecium is what type of organism? It is holozoic. Option 1 is the correct answer. 164. Mycelium of mucor rhizopus is, when you see the rhizopus, the mycelium, it is cenocytic. Both in case of mucor and rhizobium. So rhizopus, right? So this, for this particular question, the option is you go with 164. Option 1 is the correct answer for question number 164. Next, 165. Which organism is used to obtain the single cell protein? SCP we call it as. So which usually we go with the yeast. We go with the yeast, a single cell protein obtained from yeast, and also you go to get from penicillium, fusarium, even go with the spirulina, are used for substitute for the protein or the food supplement. 1 is the correct answer. Mycorrhizae works where? Mycorrhizae works in the root hair in the adverse condition, because mycorrhizae is a symbiotic relationship, right, where you could see between the fungi and the root hairs. Okay, so option 2 is the correct answer, only during adverse condition. Question number 167. Penicillium antibiotic which affects bacteria because it blocks what? It's the peptidoglycan synthesis. The bacterial cell wall is made up of peptidoglycan. So penicillium, which stops or inhibits the peptidoglycan formation. So option 1 is the correct answer. 168. Life was originated in water, first statement. Statement 2. Water remains liquid between 4 to 90 degrees centigrade. Both statement 1 and statement 2 are true, but statement 2 is not the correct explanation of the statement 1. The reason that you could see, because water is the most abundant substance in living beings. Water is the only molecule that you could see exists in liquid state at wide range of temperature. You could see like 4 to 90 degree centigrade. But water has the maximum density of 4 degrees centigrade. It freezes at 0 degrees centigrade and boils at 100 degrees centigrade. So life was originated in water more than like 3 billion years ago by aggregation and interaction of these organic molecules of what? Monosaccharides, polynucleotides, amino acids, lipids, nucleotides. We could see like polynucleotides and proteins. So option 1 is the correct answer. 169. Bacterial cell divides in every minute. It takes 1 hour to fill up a cup. How much time will be taken to fill half a cup? This is 59 minutes because the number of bacteria become doubles after each division. So option 2 is the correct answer. 170. A combined solution of copper sulfate and calcium hydroxide, which is used for the fungicide to kill the fungal species. It's a Bordeaux mixture. Bordeaux mixture is the first fungicide to be discovered. It was discovered by Professor Millardet. Professor Millardet, 170. Professor Millardet, he first discovered Bordeaux mixture. This Bordeaux mixture which used as a fungicide, which used for killing the fungi. Okay. This mixture is also called the holy water for plant physiology and also for the effective against white rust, mildews and the blights. Option 3 is the correct answer for 170. 171. The transfer of DNA from one bacteria to another is known as conjugation by means of pili. Okay. So question 172. You see the two statements, hibernation, aestivation are the short term adaptations. Aestivation is a state of remaining inactive during winter in a warm place. For this, statement 1 is true, but the statement 2 is false. Aestivation is not the state of remaining inactive during the winter. It is state of being active in the winter season. And also the aestivation where you could see like hibernation is what usually you do it, but aestivation is like going to the dormant stage. So it may not be remain active. Fine. So for this question 3 is the correct answer. Question number 173. Coming to this question 173. 173. Triple antigen or DPT is meant for a vaccine against tetanus, whooping cough. The first option is the correct answer for 173. Question number 174. Algal virus are known as, we know the different type of viruses. We have bacteriophages, we have lytic cycle, lysogenic cycle, and we have transduction also we would have studied, like generalized transduction, specialized transduction. We have different types of phages we have, the qualiphages, we have different type of phages that you would have studied. But the phages, which is actually the algal phages, right? This we call it as which affects the algal species, we call it as phytophages. Phytophages is the correct answer for question number 174. Question number 175. When a bacterium is provided with flagellum arising in two opposite ends, it is called as, we discussed like the types of flagella like amphitrichus, lophotrichus, peritrichus. Different types are based on the arrangement of the flagellum. For this question 175, where you could see the bacterium which provides the flagellum arise from the two opposite ends, where you could see this end as well as in this end. Both the ends you could see the presence of the flagellum. So this we call it as an amphitrichus, just one flagella. If you could see the tuft of flagella, then it is called as lophotrichus. You need to be very clear, one flagella means it is amphitrichus. If it is tuft of flagella, then it is called as lophotrichus. In general, which you could see in both the sides, if the amphitrichus, the option is there, you go with this option. So for this question, you go with the first option. Question number 176. Metazoans without tissue grade organization, that we call it as parazoa. The first option is the correct answer because there is slow coordination between the structures. 177. Looking at both the statement, DNA is the storehouse of all the hereditary information in eukaryotes. Statement 2, DNA is capable of replication. For this, both statement 1 and 2, of course, these are true. And the statement true is the correct explanation. So there is no just deviation within the statement. We go with option 2. Question number 178. Mycoplasma hominis. We discussed already about mycoplasma, which cause PPLO, that is pneumonia like organisms. And this is cell wall bacteria, and this is gram negative. And this causes what? This causes infertility in the males. It won't cause brain fever because TB, smallpox by virus, tuberculosis by mycobacterium, brain fever is caused by the rickettsia species, but infertility in males is because of the Mycoplasma hominis. Option 4 is the correct answer. 179. Myology is a study of muscles. Statement 2. Sarcology is a study of the muscles. Looking at these two statements, we go with the first option. Both statement 1 and 2 are true, but statement true is not the correct explanation of the statement 1, because myology or sarcology, both that we call it a study of the muscles, but it's not seems to be relevant, so it's not linked with the statement 1. The last question, question number 180. How many bacteria are produced in 4 hours if a bacteria divides once in a half hour? Just 1 to 2 minutes, 3 to, like if you just keep on 30 minutes, you just keep on accumulating. 1 will become 2, 2 will become 4, 4 will become 8, 16, 34, 62, it keeps on increasing. But they ask once in half an hour, then 4 hours when you calculate, this comes 256, because each time it's getting divided, every 30 minutes dividing into 2, 2, 2. But end of the 4th hour when you calculate, this comes around 256 bacteria get completed. By this we are completing this particular chapter, the living world. Thank you for joining with us. Thank you so much.